Good morning, microgeniuses. This is Brian, and this is the first video lecture for the class that is normally scheduled for Thursday, October 5th, 2017. And I will be lecturing and finishing Chapter 5. Then, after I'm done with Chapter 5, I will go through critical thinking and clinical applications for Lab Exercise 12. And then we'll move on to Chapter 6. Okay, and where we had stopped in lecture on Tuesday, we're talking about these different types of medically important protozoa. And so you see among these we have flagellated parasites like Giardia and Trichomonas. We have amoebas like Entamoeba histolytica is pathogenic. We have Ciliophora, which are ciliates. And the majority of ciliates, this would be like Paramecium, are harmless. And then we have Apicomplexa, which are sporozoa. And this includes some very important infections like Plasmodium, which is malaria, and Toxoplasma gondii, which is toxoplasmosis. And here is Giardia, and you can see that it is Lophotrichus. I had to think Lophotrichus. And this is Trichomonas here, and you see that they're flagellated. This is Entamoeba, and notice it doesn't really have a blobby amoeba shape in this particular slide, but it can form pseudopods and move around. And what it does is it will cause a bowel blockage, where you get amoebae and inflammatory cells, and that will completely block the intestines. It will cause constipation, diarrhea. The blockage will have to be removed surgically, and it's very, very difficult to fight this type of protozoan parasite. So lots of anti-parasitic medicines will be used, and this will be a disease state that will persist for several months. Here's Plasmodium. Plasmodium is the parasite in malaria, and here are the symptoms of malaria. And what you see here is just a sort of a background of white blood cells and then infected cells of Plasmodium. There isn't really any free Plasmodium in this slide, unfortunately, but there are lots of different symptoms for malaria. Here are a few of them: nausea and vomiting, dry cough, fever, chills, enlargement of the spleen, and it can, if it persists without any type of treatment like quinolone or quinine, then it can lead to organ failure. Okay, this is toxoplasmosis. This is Toxoplasma gondii. And again, when a woman is pregnant, should not be handling cat poop, shouldn't be touching the litter box. That's for somebody else. Cats are carriers of toxoplasmosis, and so you want to avoid any contact with their poop. Okay. Now, meiosis, we should talk about a little bit. This is sort of an odd place to put meiosis, but you start out with a zygote, which is 2N. Okay, it has 2 sets of chromosomes, and then it goes through 1 cell division. And before that cell division, you get DNA crossover. So the red chromosomes originally came from mom. The blue chromosomes originally came from dad. So there is DNA crossover. So you can have individual chromosomes that are essentially hybrids between mom and dad. That enhances genetic variability. Even without genetic crossover, when you do have meiosis and then fertilization, it lends itself to a huge level of diversity. But crossing over where you get hybrid chromosomes it doesn't necessarily happen to every chromosome during replication, but it is a feature where there's a blending effect that mom's traits and dad's traits are on the same chromosome. In cell division number 1, you still have diploid cells because the cells have produced sister chromatids. Okay. In cell division number 2, now you have single chromosomes, so they're haploid cells, and these cells then go on to mature to become mature gametes. Okay, these are free gametes, both sperm and egg, and lots of protozoa will reproduce in this particular fashion. Okay. And then after 2 gametes are formed, they undergo a process called syngamy, which is fertilization. When 2 gametes come together and form a zygote, then that we just refer to that as syngamy. Some protozoan pathogens that we have not discussed before are trypanosomes. Trypanosoma brucei causes sleeping sickness, which is prevalent in Africa. Trypanosoma cruzi produces Chagas disease. Again, and these are they're characterized by a certain level of paralysis because the toxins are paralytic, and these occur through bites with blood-sucking insects, and the insects are vectors. The insects do not get the disease, but they carry the disease, so they will transmit the disease from human to human. And here is one of the types of vectors that a type of flying bug that transmits trypanosomes. The first slide is sleeping sickness, and you see some paralysis on the eyelid. And then the second is the heart, and this shows damage, organ damage to the heart after a patient has died from Chagas disease. And you can see that all the chambers, they just basically look like threads where the parasite has just eaten away through the chambers of the heart. Entamoeba causes amoebic dysentery, or we can call this amoebiasis, and this is caused primarily by Entamoeba histolytica. And this is transmitted from human to human, and it's almost always associated with humans. You can get it primarily through contaminated water, and it is a protozoan that forms cysts. Cysts, again, are the dormant state for these parasites, and trophozoites are the vegetative state. So please know the difference between a cyst and a trophozoite, and these are associated with disease. Obviously, the cyst, under a lack of nutrition or harsh environment, then you get the cysts in food and water. They go through the stomach, and then they get down into the intestinal tract, and here, and it's nice and yummy. And the trophozoites are released from the cysts in these yummy portions here. I lost my arrow. Okay, there's my arrow. And the mature trophozoites then transit through the large and small intestines. And then when they hit the anus, there's less nutrition, and cysts form again. Cysts pass through the feces. The feces contaminate water, they contaminate food, and the whole cycle continues. Okay. Now, nobody likes worms. So we're going to talk about worms. There's a lot of diseases that are caused by worms. There are 2 types of worms. The first one is called flatworms. They're very thin, and they have a segmented body plane. And the first one is cestodes, and cestodes is characteristic of long, thin worms that look like ribbons, like tapeworms. And termitodes would include flukes, which have flat ovoid bodies, and I'll show you some pictures of this in just a minute. Then in contrast to roundworms, these are elongated and they have unsegmented bodies. This would include nematodes. Nematodes are associated a lot with plants, and in legumes, leguminous plants like beans, you'll see a lot of nematodes that will form in the nodules of the plant stem. But there are infective roundworms as well. And here is just You don't need to know all these body parts. You don't need to know the physiology of a helminth, but you'll see there is a lot of physiology that is similar to humans. There's a lot of physiological features. There's a separate mouth and anus. There are male and female parts. Um, and so these become difficult to treat because their physiology is so similar to humans that if you come up with a therapeutic or some type of drug or anti-helminth drug that will work against these worms, they'll also cause the same effects to the human body. So you have to be very, very careful with that. It's difficult, again, because their physiology is so similar to humans that you need to come up with some type of anti-helminth drug that's not going to affect human physiology. Okay, here's a fluke. The fluke then, I believe that their mouth and their anus are the same thing. And so they eat through their mouth, they digest the food, and then they excrete the waste also through their mouth. Okay, just in separate cycles. Okay, here's some flukes. They're so cute. You can see their little eyes. They can bend and wriggle and move, so they have locomotion. And here is a roundworm that has been taken from I believe this is a dog. Okay, you can see the roundworm is coming out of the intestines. Okay, and helminths are hatched from eggs. You know, they're very similar to humans. They have eggs. The eggs do not. The eggs dwell outside of the helminth body, unlike humans. So they have eggs, larval, and adult stages. And the majority of the helminths are sexually reproduced in the host body. In nematodes, those are roundworms, the sexes are different with unique gender features. In trematodes, the sexes can be separate or they can be hermaphroditic. Hermaphroditic means that the same individual has both male and female parts. And then cestodes are generally hermaphroditic. Remember, trematodes and cestodes are flatworms. Okay. And so in this particular instance, the egg and larva can be transmitted to the same host, can be reinfected, or they can be infected to a different host. Okay. The larval development occurs in the intermediate host, and the intermediate host can or cannot, may experience symptoms at this point or may not. And then adulthood and mating occur in the definitive or the final host. And these hosts, the intermediate and final host, it can be the same person that is basically just cultivating these worms throughout their life cycle, or they can be transmitted from person to person where you have a different intermediate host that is developing the larva, and then the final host is developing the adult and then holding a little sex party so they can mate. Okay, so they can be the same or different. There is a vector, and it can be a vector. Usually this vector is a physical vector. It's not, something that ingests it, but the vector is the intermediate host that suffers no ill effects. When a vector by definition carries around the pathogen, but doesn't suffer any ill effects. Okay, and interestingly enough, the mating cycle for helminths is lunar. And so you hear all this business about people going a little wacko during the full moon. It could be that in their intestinal tract, they're hosting a little sex party during a full moon because these helminths come out, they burrow into the intestinal wall, and they come out of the intestinal wall for mating. And this happens around the same time that there's a full moon. Don't ask me why. I don't know why these helminths know that there's a full moon, but it is a lunar cycle. Okay. Now, pinworms is interesting, and it's indigenous to California. It's more indigenous to Southern California because of the climate, but almost every school child gets pinworms at least once in their lifetime. And so here's a physiology of a pinworm. Okay. And a pinworm is more of a flatworm. It's not round. Okay. And here's the perianal region, and you see these little white things or pinworms in that particular region. Usually the way that pinworms are diagnosed is you just take a piece of Scotch tape and then you just adhere the tape to the perianal region, rip it off, and you'll get some worms on the tape. And you show the worms to the doctor, and the doctor says, oh, yes, you have pinworms. And so here's an individual who ingests some embryonic eggs, okay, digging in its bottom, no hand washing, goes from anal to oral. Larvae are hatched in the intestines. They penetrate the intestinal mucosa, and when they penetrate the intestinal mucosa, they encounter all these polysaccharides and proteins that are super yummy, and they mature in the small intestines, in the upper colon. They mate in the colon in small intestines, and then pregnant worms, this says gravid worms, gravid is just the same thing as pregnant. They migrate to the rectum, and then they lay their eggs in the perianal region. And here are some gravid worms, and they are just laying their eggs there. Okay. And there are about 50 parasitic helminth species known to infect humans. Helminths do not survive well in cold environments, so you see them in tropical regions. And they co-migrate with human hosts. So if somebody has an infection of pinworms and they go into a colder region, then they can co-migrate. You can have an outbreak in a colder region because the human hosts are carrying them and are acting as like pseudo vectors as they migrate. Okay, there's about 50 million helminth infections per year, which is about 1/6 of the population. And the primary target is malnourished children. And so malnourished children are also immunocompromised because when you have a poor diet, then your immune system suffers. And so they have a harder time fighting off these infections. Okay. So let's review. So 2 types of fungi. We went over these, and the 2 types of fungi are molds and yeast. 2 types of protists. Remember, protists and fungi are kingdoms in the domain Eukarya. And so the 2 different types of protists would be the 2 subkingdoms, algae and protozoa. What do algae have that protozoa don't? Algae have chloroplasts. Okay, algae are photosynthetic, they're phototrophic, and protozoa are heterotrophic. Phototrophic means that the algae can make their own organic carbon, and heterotrophic means that the protozoa have to get their organic carbon from other sources. What is a cyst? And this is in terms of microbiology. And you'll remember that there are 2 states of specific protozoa. Not all protozoa do this, but there are 2 states. One is called the trophozoite, and the trophozoite is an active vegetative form of the protozoan, and a cyst is the dormant form of the protozoan. Amoebas will form cysts, and when amoebic cysts get into the water supply, then specific species of amoeba can cause diarrheal disease. Okay, but the cysts are dormant under harsh conditions, low nutrition, low water, and the trophozoites are vegetative. Once the cyst is rehydrated and introduced into an environment with nutrition, then it becomes a trophozoite. The definitive or final host for helminths, that final host is where the helminth actually it becomes an adult, so it allows for the maturation of the helminth to adulthood, and the definitive host is where mating occurs. And so after mating occurs, the pregnant helminths then migrate to the perianal region, and that's where they lay their eggs. Okay, and so look at that. We finished chapter 5, so we're done with eukaryotic cells. Not completely done. We're going to review this over and over again. Let's see, we have 2 pages of glossary, so make sure that you look over the review slides and the glossary. Okay, Next, I will discuss critical thinking and clinical applications for exercise 12. So this is page 106 of your lab textbook, okay, exercise 12. In critical thinking, what ingredient makes mannitol salt selective? Okay, well, first of all, let's look at the two ingredients that are listed here. These are pretty key. Mannitol is just a sugar. And so I don't think, most microorganisms, most bacteria can metabolize mannitol just the way that they would metabolize glucose. So I don't think it's selective. So that leaves salt. And highly, a high concentration of salt is indeed selective. It selects out for organisms that would have a thicker cell wall because they can handle better the osmotic stress that would be associated with a salty environment. And if you think of the different bacteria that are cultivated on your skin, like Staphylococcus aureus and Staphylococcus epidermidis, your skin does secrete salt, and along with oils and along with water and sweat, you do have salty skin. And so this would select out specifically for gram-positive bacteria. Okay, and then for number two, you should be able to fill in the blanks on your own. And you should also note which bacteria are gram-positive and which bacteria are gram-negative, okay? And so make sure that you either circle them and put a plus sign next to them or a negative sign, or do something that when you filled in these four blanks that I know which ones are gram-positive and which ones are gram-negative. Okay, then the clinical applications. Skip. These are they're really hard to answer. I just skip them all. Skip them. Skip like the wind. I don't really think that it's necessary that you look these up. What you're going to find is that antibiotics are selective, and they will select out different bacteria that are antibiotic resistant, and they will not allow the growth of those that are susceptible to antibiotics. And so that's what you're seeing. And as long as you know the difference between selective and differential media in this particular exercise, then I think that you've learned a lot. And I do want you to remember that you saw instances of selection as well as differentiation. In mannitol salts media, the salt was selective for only gram-positive organisms, and the mannitol was differential because certain bacteria, namely Staphylococcus aureus, would ferment mannitol and turn the media yellow. Everybody saw that. So you have a selective aspect of mannitol salts agar, and you have a differential aspect of mannitol salts agar. EMB agar would only select for gram-negative bacteria. Okay, so it was selective. But you also saw that an EMB agar, if you grew E. coli, that E. coli would grow with a metallic green sheen. Okay, so it was also differential because you could differentiate in this case from E. coli and any other gram-negative species of bacteria that happen to grow an EMB agar. So when you're asking which media are selective, both are. And which media are differential, both are. They both have aspects that cause them to select out bacteria and to differentiate between the colonies of bacteria that end up growing on each of the media. So anyhow, I hope that is very helpful to you. We'll now move on to chapter 6. And chapter 6 is all about viruses. Now, viruses are very different. We don't refer to viruses as being alive or dead. We refer to them as being active or inactive. In terms of life, viruses stand out among the other forms of microorganisms because the type of life that they have is very different. They're by and large parasitic, pathogenic, or parasitic because they require a host. So they have to have a host cell in order to survive. Their physiology is very similar. On their own, when they're non-infective, they lay in a dormant state. After a while, they will inactivate in that dormant state. But if they are picked up by a host, then they can be reactivated and they can hijack the host machinery in order to propagate the virus. So our objectives here are to understand how viruses fit in on the biological spectrum. They're non-cellular life. We don't call them prokaryotic or eukaryotic because they don't have cells, but we need to understand how they fit in biologically. We want to look specifically at virus structure. Virus structure is much more simple than what you would find even a prokaryotic cell. And we'll talk about the classes and names of the different viruses, understand how they multiply, and understand virus culture, and even talk about medically important viruses. There's obviously a lot of viral diseases. Some are rather insidious. Influenza is a virus. A lot of the what we would call quote unquote childhood diseases are viral. Hepatitis is viral. And so there is a really, really strong interest in being able to both prevent viral disease as well as treat viral disease. Okay. Are viruses alive or dead? Okay. We usually refer to them as infectious particles and not living entities. And rather than saying it's alive, then we'll say it's active. And rather than saying it's dead, we'll say that it's inactive. Okay. And interestingly enough, we're finding out as scientists that the human genome is very, very fluid. And the genome is all the genes, all the chromosomes, everything that is in the human body. And scientists think that up to 90% of the human genome actually integrated into our genome from viruses. So viruses play an important role in terms of donating their DNA to other organisms and allowing that DNA to be incorporated into other organisms to confer different traits. Okay, so this is just a table from your book. They're obligate intracellular parasites. Okay, they're parasitic. Viruses infect everything. There are even viruses that infect bacteria. They are everywhere. They're ubiquitous in nature. Ubiquitous just means that they're extremely prevalent and they have a major impact on human life and all biological life. They're very, very small. Some viruses are down to 20 nanometers. The largest virus perhaps is a smallpox virus, which is about 450 nanometers in diameter. You can almost see it with a light microscope. They are not cells. They do not have a cellular structure. They do not have a plasma membrane. And they do not independently fulfill the characteristics of life. You can't just culture viruses and have them replicate without a host cell. Okay, and so they're active inside host cells and they're inactive outside of the host cell. And their basic structure consists of a protein, which we call a capsid, and the capsid is the outer portion, and that surrounds a nucleic acid core. Nucleic acids can be either DNA or RNA, but we do not have both in the same species. DNA can be double or single-stranded, RNA can be double or single-stranded, and these viruses, the molecules on the surface of viruses, which are called spikes, are very specific to the cells that they infect. Okay, so they can look at a host cell like a human cell look at the proteins on the host cell, and they can attach directly on that particular host. Okay, the virus, if it's human infective, is not going to get confused and attached to bacteria. You have high specificity, and so that virus has adapted to be able to very specifically find its host. Okay, they multiply by hijacking the cell's genetic material and regulating the assembly and synthesis of new viruses. What they do is they simply use the cell's RNA polymerase in order to transcribe genes. Then once the genes are transcribed, the viral genes as messenger RNA are translated by the host cell's ribosomes to make the virus proteins. Once the virus proteins are then synthesized, then they self-assemble to make new viruses. Okay. They do not accomplish most metabolic processes, and they lack the machinery for synthesizing proteins. So they can't synthesize their own proteins. They can't synthesize proteins unless they've hijacked a host cell. Okay, so let's break down the viral components. First, you have some type of covering, and then you have a central core. And the outermost covering is made out of protein. It's called a capsid, and you'll find a capsid in all viruses. And then inside the capsid, you will find, outside of the capsid, you will find an envelope. So I screwed up, sorry. Capsid is on the inside, the envelope is on the outside, and the envelope is not found in all viruses. There are envelope viruses, and then there are naked viruses, okay? And then the capsid and envelope protect the central core, which has nucleic acids, and can be a DNA virus, can be an RNA virus. And then the central core also has matrix proteins and some enzymes that are sometimes found in viruses. The matrix proteins function just to provide structure to the nucleic acids, and the enzymes may provide some type of metabolic function, but they tend to be rare in viruses. Okay, so you can have an envelope virus that's B, that has a covering, just a phospholipid bilayer, and then you can have a naked virus, and the naked virus just has a capsid in it, and the capsid is kind of a polygon shape, and the polygon shape is protecting the nucleic acid inside. At the apex of every one of these capsid portions, you find a spike, and these spikes are glycoproteins. They contain sugar and protein, and they're highly specific for attachment to the host cell. You find spikes on the envelope as well in envelope viruses, and the capsid does not have any spikes. Okay, so the capsid is made out of protein components called capsimers, and these are just subunits, usually a single protein, and the subunits self-assemble to make a finished capsid. And you can have a helical capsid. A helical capsid looks more like a cone when you actually look at the virus, but inside the DNA is making a helix. And then you also can have a big polygon that looks like it approximates a sphere, and that is icosohedral. Helical capsids, like I said, form a continuous helix, and the nucleic acid is coiled in the helix. Helical capsules, this would be like tobacco mosaic virus, which kills tobacco, influenza, measles, and rabies. Okay, so a helical capsid looks like this, and the capsimers are those small things that look like capsules that you would take, like a Dristan capsule, looks like medicine. And then as the capsimers assemble, then they will protect the nucleic acid in the inside, and the capsimer will then develop a full capsid that protects the entire nucleic acid. Here's an envelope virus, and you see that there is a capsid on the inside that's protecting the helical structure of the DNA, and then an envelope on the outside. Okay, here's a helical virus, and the helical virus is just a capsid, it's just a capsid made out of capsimers. The envelope virus has a helix in it, and the helical virus obviously does not have an envelope, but it is forming a helical structure that just basically looks like a cylinder. And so these envelope viruses here, you can see kind of a little helix inside of each one of these envelopes, but the predominant feature is the envelope, which is more of a blob, a little more circular. And then these helical capsids, then they just look like cylinders, look like little grains of rice, but they're actually helices that have been formed, and the outer structure just approximates a cylinder. Icosohedral capsids. This is a 20-sided figure, and the 20-sided figure has 12 even-spaced corners. And you can have a single type of capsimer. This would be 20 capsimers, or you can have different types of capsimers. You can have a variety in the same virus. And the capsid will appear spherical. Some of them appear cubical. Here's an icosohedral virus with no envelope, and you can see that. And here's one that this is just a protein diagram. It's not an actual picture of the virus, but it shows that it approximates a spherical shape. So we have helical capsids, we have icosohedral capsids, and then we can have what are called complex capsids. Complex capsids have multiple types of proteins, and they have asymmetrical shapes. Okay, so this is the third type, sort of a catch-all type, where we can't, if a capsid doesn't look icosohedral or helical, then we just throw it into this complex category. Okay, this is an interesting virus. It's called a bacteriophage. Phage is just another word for virus. Bacteriophage implies that it infects bacteria, and it has an icosohedral head. Okay, that's where the DNA is contained. But here then it has a neat delivery mechanism. So these heads will actually then hang off of the end of the virus, and these tail fibers and this base plate will adhere to the surface of the bacteria. And then the sheath allows for injection of the viral DNA directly into the bacteria. Okay. So, and I'll show you a picture of this. This is kind of a cool process. The icosohedral head never actually makes it into the bacterial cell. It doesn't need to. But the rest of the virus acts like a delivery system, and it just simply injects the DNA into the bacterial host cell where more viruses can be formed. Okay. Like I said, all viruses have capsids, but not all viruses have envelopes. Okay. In this case, the virus actually takes some of the membrane. This could be the cellular membrane, or it could be a membrane from one of the organelles from a host cell. Okay, and these envelopes are generally envelope viruses infect animal cells. And then the virus, after it hijacks the membrane, actually takes the host cell's membrane, then it will sort of eject the membrane proteins from the host and replace it with its own membrane proteins. And so the viral envelope will be covered with these sort of protein spikes that are indigenous to the virus, where the membrane itself was indigenous to the host cell. Okay, these proteins are called spikes, and they're glycoproteins. That means that they're part glucose or part sugar and part protein. And they protrude out from the envelope, and they allow the virus to attach directly to the host cell. Okay. And envelope viruses are pleomorphic. That means that they're shapeshifters, and so the envelope can take on different shapes depending on the application. It can move around. It can move to accommodate injection of the viral DNA into the host cell. Okay. And you'll see that envelope viruses can have helical capsids, or envelope viruses can have icosohedral capsids. Okay. And it has different glycoprotein spikes. Okay. Here's an example of a virus with a helical capsid. And you can see the glycoprotein spikes here. And then here's some envelope viruses with polyhedral capsids. And if you look at the outer covering of the virus, its primary purpose is to protect nucleic acid. Excuse me. This functions when the virus is outside of the host cell. And so you want, if you're a virus, to be able to be protected during harsh environments. And so you see these capsids and envelopes in enteric viruses, because the virus has to survive the stomach, it has to survive the duodenum and the intestines. And so these viruses are protease resistant. They are very, very tough, and they'll survive the digestive system until they actually find in the lower portion of the intestines and the colon a place to infect. Okay. The other function of the capsid envelope is to be able to bind the cell surface, the host cell surface, with special binding proteins. These are the glycoprotein spikes will bind directly to the receptor proteins of the host cell. Okay. And then they'll assist in the penetration of the host cell and allow the viral nucleic acids to actually go into the cytoplasm of the host cell. And the coat proteins can also stimulate the immune system. The immune system, obviously, when you're infected with a virus, is going to be alerted. And so the coat proteins are antigenic. They do not look like human cells. And so that's going to alert the immune system. And then by stimulating the immune system, the body can mount an immune attack against the virus. At the inside of the virus, we have nucleic acids. Viruses are either DNA viruses or RNA viruses, but they're not both. Both encode genes, and so both are highly appropriate. You don't necessarily need DNA if you have messenger RNA, and so therefore these viruses have adapted to only have ribonucleic acid rather than deoxyribonucleic acid. If you look at the virus genome, the number of genes is quite small. For hepatitis B, there's only 4 genes, so with 4 proteins it can survive. Herpes virus is much more complex. It has multiple genes. And then if we have single-stranded DNA, those are called parvoviruses. Double-stranded RNA is called a reovirus. And these are important terms to remember. Single-stranded RNA are ready to translate into protein. So single-stranded RNA that are positive sense, the ribosome of the host cell can just attach to that positive sense RNA, and it can start to make the viral proteins. Okay. Negative sense RNA has to be converted to positive sense RNA by the host cell, like an RNA polymerase or a reverse transcriptase or something like that, will translate the RNA into positive sense RNA, and then it can be converted into protein. Okay. And here are a lot of different classes of viruses. These are relevant to DNA only. Okay. And I'll tell you in these, there's a lot of terminology, and you don't need to know all of these. You should know the different classifications, you enveloped or non-enveloped. And under enveloped viruses, you only have double-stranded DNA. In non-enveloped viruses, you have both double-stranded and single-stranded DNA. Okay, and namely, it gets a little tough because like single-stranded genomes are all called parvoviruses, but then double-stranded genomes, you have adenoviruses are linear, and papovaviruses are circular. And then if it's a double-stranded genome in an envelope virus, a pox virus is helical capsid, and a herpes virus has an icosohedral capsid. So out of this, I would want you to know the broad classes, and I will be very, very specific on the review sheet as to which viruses you should know, which are the most important. I would certainly know parvovirus and reovirus at this particular point in time. Okay. RNA viruses, it becomes much more complex. There's non-enveloped and enveloped RNA viruses. If you look at the non-enveloped, there are double-stranded genomes called reoviruses. There are single stranded genomes that are picornaviruses and caliciviruses, and then in RNA viruses, enveloped viruses are all single-stranded genomes. And in the envelopes, you have a specific type of virus. You should know this type of virus, circle it. It's called a retrovirus. And this single-stranded genome that encodes reverse transcriptase, this retrovirus, it actually can make DNA from its RNA because it encodes reverse transcriptase, and the DNA can integrate into the host genome. This is medically relevant because it includes HIV. HIV is a retrovirus. HIV virus can integrate into the human genome and can be carried forever. In terms of single-stranded genomes without reverse transcriptase, you have segmented and non-segmented genomes, and you don't need to know these different classes of segmented and non-segmented genomes. So, I would know what a reovirus is, I would know what a retrovirus is, and I would know the different classifications. Okay. In viruses, there are 3 orders. There are 73 families, and there are 287 genera. Okay. And so the orders are Codovirales, Mononegavirales, Nidovirales, and you should know the different hosts that are associated with these different orders. Codovirales are bacterium, so these are bacteriophage. Mononegavirales and Nidovirales include plant and human variants. Mononegavirales includes measles and Ebola, and Nidovirales includes rubella. And so these are medically important viruses, so I would know those. Okay, and usually we don't use binomial nomenclature for viruses. Usually instead we just use either the genus or the common name. For the genus for measles is Morbilla, genus for Ebola is Ebolavirus, the genus for rubella is Rubavirus. And so I would know, for these common virus species, I would know their genus as well. At this point, I am going to conclude the first lecture. We're at 48 minutes. I apologize, I went 3 minutes over. So the next lecture will be a little shorter. So this concludes video, or video, I guess, lecture number 1. This is the second video lecture for microbiology, dated October 5th, 2017. We're in chapter 6, and we're talking about viruses. And in terms of how viral cells multiply in animal cells, this just gives you a brief overview. And I did something wrong there. I called a virus a cell. And if you'll note from what I had said earlier, viruses are not cells. And so therefore, I need to talk about them as virus particles. Okay, the first step is adsorption. It's not absorption, but adsorption. Adsorption means that if something soaks into the surface of something else, then it is adsorbed. It is a surface phenomena, and it does not, absorption applies that the phenomena goes below the layers of the surface. Adsorption implies that it is only involved with the surface. Then after adsorption, there's penetration. where the viral nucleic acids penetrate the cell and go into the cytoplasm. At that point, the capsid is uncoated, and that exposes the viral nucleic acids. Then the nucleic acids can be synthesized, as well as the protein coat, the capsid, and all these components. After they're synthesized, then they're assembled into mature viruses. And then after the viruses are assembled, they're released from the host cell. And since there are different types of viruses, we have to consider how this is done in RNA viruses, in DNA viruses, in envelope viruses, and in non-envelope viruses. Okay, and so this just gives you an overview. We're starting out here with a positive sense RNA virus. Okay, the positive sense RNA virus is encapsulated with a membrane. Okay, so it does have a membrane, and the cell membrane interacts with the glycoprotein spikes. Okay, the cell membrane has receptors on it that these glycoprotein spikes on the virus recognize. And when these two proteins come together, then that allows the virus entry into the cell. Okay. The receptors literally wrap the entire virus, okay, and the virus then penetrates the cell membrane, goes into the cell where it's uncoated. Okay. When it's uncoated, then it is free RNA. This is, like I said, it's positive sense RNA. And then positive sense RNA can do several different things. It can go into the nucleus. I'm sorry, it stays in the cytoplasm where it becomes copies of negative sense RNA. Then that negative sense RNA then becomes, I lost my arrow, I hate that. Okay, negative sense RNA then can provide additional copies of positive sense RNA. Okay, so these are just little virus nucleic acid particles here. The RNA then can also be translated to make protein, and the protein is in the form of capsimers. The RNA can also be translated into spikes, glycoprotein spikes, that will allow for recognition of the virus to the host cell surface. And then all of these are assembled. The capsimers are assembled into capsids, and the capsids cover the positive sense RNA. The glycoprotein spikes will replace the cell's glycoproteins in the cell membrane, okay, here. And then the cell will, the virus will essentially bud on the cell membrane, take a portion of the cell membrane with it, with the new glycoprotein spikes that are virus specific, and it will be released. Okay, this is a good slide to study, so you know all the different parts in terms of how a cell that is encapsulated, that has a membrane around it, or excuse me, how a virus is encapsulated, goes into the cell and is replicated. And you should note that this is a positive sense RNA virus. That's important to note because the RNA to be replicated does not have to go into the cell's nucleus. Okay, and this just shows you another non-encapsulated virus, okay? Or I'm sorry, this is an envelope virus. Wrong, Brian. Okay, this is does have an envelope around it, and how it interacts with this host cell membrane, and the glycoprotein spikes are then specific to receptors that coat the cell membrane. Okay, slide B shows a non-envelope virus, and the capsids have spikes, and the spikes again are specific to the host cell membrane, and this relationship between the capsid spike and the host cell membrane receptors allows the virus to invade the host cell. Okay. You can have specific attachment, and then engulfment. where the entire virus is taken into the cell in a vesicle, okay? Or you can have attachment, what's called irreversible attachment, where the two membranes fuse, the virus membrane as well as the cell membrane fuse, and then the cell membrane becomes a part of the virus membrane or vice versa, and the nucleocapsid, the capsid itself, is then released into the cell. So it can happen either way, okay? This happens to show this on a DNA virus. This happens to show this on an RNA virus. It can be vice versa. This type of attachment in letter B in the figure isn't specific to RNA, and the type of attachment in A in the figure isn't specific to DNA. It's just showing two different ways that the virus can get into the cell. Now, DNA viruses, in order to replicate their nucleic acid, this is a process that only occurs in the nucleus of the animal cell. Okay, so the DNA viruses have to enter the nucleus, and then the DNA portion is replicated by synthesizing within the nucleus. DNA is replicated using an enzyme called DNA polymerase. DNA polymerase only exists in the nucleus of eukaryotic cells, and so therefore the virus can only replicate if it goes into the nucleus. RNA viruses are replicated using RNA polymerase, and RNA polymerase exists both in the nucleus and the cytoplasm. So RNA viruses are replicated and assembled in the cytoplasm. Okay. And this shows you some different routes in terms of how different viruses, this is for positive single-stranded RNA, negative single-stranded RNA, negative or double-stranded RNA, and then retroviruses, the 4 different types of RNA viruses. This shows how the RNA is replicated. Okay. Positive RNA virus is converted to negative RNA. That negative RNA is then replicated and it becomes a part of the viral nucleic acid. And that negative sense RNA then can be encapsulated into a capsimer here, or into a capsid here. Okay. Negative strand RNA viruses can be converted into positive sense RNA. They then can go into the genome, or they can become negative sense RNA where they're packaged into an encapsulated virus. Double-stranded RNA viruses will make positive sense RNA. Those can be incorporated into the genome, okay? Or they can go further to make double-stranded RNA, and then double-stranded RNA, which is a reovirus, is captured then by the capsid. Okay, retroviruses that have their own reverse transcriptase. In retroviruses, single-stranded positive sense RNA makes single-stranded negative sense DNA. Then the host cell at that point in time will make the positive sense strand, so you get double helix DNA. Double helix DNA can incorporate into the host cell's genome, okay? Or it can make more retroviruses. Okay, retroviruses like HIV are particularly insidious because they can make double-stranded DNA, and double-stranded DNA can incorporate into your own genome, and it's very difficult to get out. Once you're infected with double-stranded DNA from HIV, then there is literally no way that you can get rid of that DNA. Okay, I don't like this slide quite as much. I do apologize for it. But what it shows is when viral DNA has to be replicated, you start out in the cytoplasm, then it goes into the nucleus. In the nucleus, the viral DNA is transcribed into messenger RNA that's translated to make viral proteins. In addition, the viral DNA is unwound and unzipped So you have positive and negative sense DNA. These then are complemented with complementary strands. Okay, these complementary strands can integrate into the host cell DNA, or they can go to the production of more viruses. The viruses are actually assembled in the nucleus. Okay, and when the virus is assembled in the nucleus, then it's released from the nucleus, has to go through the cytoplasm, and then finally be released from the cell. Okay. And when double-stranded DNA inserts itself into host DNA, we call that latency. So you can have a latent virus, and that way, lots of viruses like herpes and HIV become latent. And when they're latent, they're essentially hiding in the host DNA. And then the host DNA can release the viral DNA particles at any point in time, and that can cause a flare-up of an infection. Okay. Now, when you have a non-envelope virus or a complex virus, this just releases when the cell lyses. Okay, remember lysis just means break apart. So when the cell ruptures, these non-enveloped and complex viruses are released. They can go and infect other cells. Envelope viruses, like we saw in previous slides, are liberated by budding, okay, or exocytosis, which is basically the same thing, from the membrane, and when they do this, they capture a portion of the membrane from the host cell. And this budding or exocytosis can occur in the cytoplasm, it can occur in the nucleus, it can occur in the endoplasmic reticulum, or in any number of vesicles. Okay, so any of these organelles or even the entire cell can donate their membrane in order to make an envelope virus. Okay. And this just shows an RNA virus, okay, with its capsid. Its capsid then covers the RNA, okay. Then matrix proteins line up with the host cell membrane. Matrix proteins then replace the host cell receptors with glycoprotein spikes, okay. Then the nucleic acid with the capsid orients itself so it's along the lines of the region of budding. The cell will start to bud a virus. At this point, we call it a virion. And then the virion, which is just a single virus particle, is released, and it has a portion of the cell's membrane as an envelope. Okay, and here's some envelope viruses that are budding from a host cell. At the same time, this causes damage to the host cell. It causes alterations to the microscopic appearance. It can cause what are called inclusion bodies, which are just bodies of compacted virus, cell debris, organelles that will occur in the nucleus or the cytoplasm. It's just a way for the cell to store dead and dying material. And it will also form syncytia. Syncytia are multiple cells that fuse together. All of a sudden, instead of having several cell membranes holding intact cells, all of the cell membranes will fuse, and it'll look like you just have a single very large cell that has multiple nuclei. Okay, here's an inclusion body in a muscle cell. Okay, it's very, very large. And then here's a large cytoplasmic mass, and you can see multiple nuclei here and here. And this is just a large syncytia where you've had many, many cells all fused together. And at this point, the syncytia is not, the cells don't function very well with multiple nuclei. So the syncytia with the cytoplasmic mass will essentially die off. Okay. In terms of viral infections, they can last a few weeks, like a common cold or influenza or something like that, or the rest of the lifetime of the patient. Latent viruses can remain in the cytoplasm of the host cell for a long time. They don't have to bud and form virions. Okay. Proviruses This is when the viral DNA incorporates into the DNA of the host cell. And there's a childhood disease that causes just a red rash on the cheeks called roseola that's a provirus. And then when a virus is latent over a long period of time, we call that a chronic latent state. Herpes can achieve a chronic latent state. Chickenpox can achieve its chronic latent state, as well as HIV. Okay, here's roseola. Okay. Here's herpes, which is, obviously this is on the labial area. And this is not a latent state of the virus. This is a flare-up of the virus. So the virus is actively dividing at this particular point in time. Okay, and here's skin herpes, non-genital herpes, and it's just forming cold sores here. Okay. Some viruses can cause cancer. Okay, so this is primarily retroviruses. You also have what are called DNA tumor viruses. And in the first slide, you've got a retrovirus that's enveloped with a membrane. The DNA is released. RNA is released. This creates viral DNA that contains oncogenes. The viral oncogenes then integrate into the genome. And then the viral proteins are oncogenic. The viral proteins will do things like eliminate tumor suppression genes or stimulate other oncogenes and cause the cell to divide and replicate out of control. Other retroviruses here will initiate a cellular oncogene in a provirus state, and the cellular oncogene, once it's turned on by the viral DNA, then will initiate an undifferentiated cell growth, and the cell will start to form a tumor. DNA tumor viruses, rather than retroviruses, are incorporating DNA, and this viral DNA will incorporate into the host cell genome, create viral proteins, again causing the cell to grow out of control, leading to the formation of tumors. And cells can actually be transformed so they grow in a tumor-like state. In the laboratory, when we transform mammalian cells, we call this immortalization, because we'll take a human cell that may survive in a petri dish for, maybe 10 days. And when we transform that cell, then we can immortalize it, and it will just grow forever. It will grow and divide and continue and grow and divide. But the way that we transform that is we infect it with a virus. These transformed cells have an increased rate of growth. Their chromosomes are altered, so they grow in more of a cancer-like state. Their cell surface molecules become more viral in origin, and their capacity to divide outside of the body and even inside of the body, they can divide for an indefinite time period. Okay. And there are certain types of viruses that are infective that cause this. These are not necessarily used in the laboratory, but they do cause a problem in terms of producing cancers. And so human papillomavirus, HPV, can cause cervical cancer. Not all forms of human papillomavirus are cancer causing, but some of them are. Herpes virus can lead to a condition called Burkitt's lymphoma, and hepatitis B can eventually promote liver cancer as the liver becomes transformed with the hepatitis virus. Okay. Viruses that infect bacteria we call phage. And most of them are double-stranded DNA, but there are other forms, single-stranded DNA and single-stranded RNA forms exist. And when phage infect bacteria, they actually give the bacteria, confer, different types of genes to the bacteria that make the bacteria more pathogenic to humans. Okay. And here is a super busy slide. Okay, I'm just gonna take a time out here and see how far we've gone. Yes, that's a super busy slide, but what the heck, let's go through it. Find it again. That's not it. Let me super busy slide go. Here it is. Okay. Hang on. And this is a bacteriophage that looks like a little lunar module from the NASA space program in the 1960s and early 1970s. Here's an E. coli host cell. E. coli is bacteria. The bacterial DNA is just in a nice little round chromosome, and the viral DNA is actually being injected directly into the bacterial cell. Now, the viral DNA can actually integrate into the bacterial chromosome as a prophage. And then this bacteria we call lysogenic because it incorporates some type of virus DNA and it harbors that virus DNA. The virus DNA can stay in the genome, it can splice out, so you have separate bacterial DNA, but a lysogenic state on bacteria, a lot of times this gives this bacteria some type of adaptive advantage, and in that advantage, it causes the bacteria to become more pathogenic. So lysogenic bacteria we have to be very careful with because they can promote disease and also promote antibiotic resistance. Okay, we go over to penetration here. The other thing that can happen is that the phage can duplicate. Okay, the phage can assemble, mature, and then lyse the weakened bacterial cell, and then the viruses are released. Okay, this is the normal fate of phage that infect bacteria, but you do get the lysogenic state in specific bacteria. Here, the bacteria are dying on this side, but over on this side, the lysogenic state, the bacteria are actually getting an advantage by having the viral genome incorporated into the bacterial genome. Okay, and here are some different types of phage. These are used primarily to infect E. coli. They're called T-even phage. Don't worry about where T2 and T4 come from, but they have an icosahedral capsid head, and then the docking mechanism, that long tube, allows delivery of the DNA. So the virus doesn't have to actually invade the cell and uncoat. The DNA with this docking mechanism is just simply injected into the bacterial cell. And then that DNA, once it's transcribed and translated, then it creates all of the different portions of the new phage. Remember, phage is just a bacterial virus. So all of these components self-assemble. Okay. And so this is a phage with an icosahedral head. It's just releasing its viral nucleic acid through the cell wall and cell membrane. Here's a really cool photomicrograph showing a bacterial cell that's being infected by multiple phage over the same time period. And all these phage are injecting their DNA contents directly into the cell. Now, when we get lysogenic bacteria, these are more temperate phages. Temperate usually means that they're a little more well-mannered, I guess, for lack of a better term. But a temperate phage is one that doesn't aggressively replicate in the host cell. It replicates at a slower rate, and they will form a prophage where the viral DNA is inserted into bacterial chromosome. And when this happens, we call this lysogeny. So lysogeny is just when the bacterial DNA incorporates the viral DNA, and then with that, the bacterial DNA gets some new trait. Okay. And this allows the virus to spread. The virus just spreads along with the bacteria, and it doesn't kill the bacterial host. Okay, in terms of human disease, there are some important bacteria where lysogeny has occurred, and that causes the bacterial infection to be more insidious. And these lysogenic portions, these actual viral portions that incorporate into the bacterial genome, contain genes that produce toxins that are pathogenic to humans. So when bacteria acquires a trait from phage, we call that the lysogenic conversion. And the diphtheria toxin from Clostridia diphtheriae, Vibrio cholerae, which causes cholera, and botulism toxin in C. botulinum are all examples of virus-based genes that incorporated into the bacterial genome. And these bacteria, once the viral DNA is incorporated, then the bacteria grow and divide, and they continue to replicate the viral gene, and that confers a new level of toxicity to the bacteria. Okay. Now, it's important to understand these animal viruses, especially those that are causing disease. So you take the clinical specimen, it could be a piece of tissue, could be a body fluid, and you identify the virus in a clinical setting. You can prepare the virus for use in vaccines. And you can also do research regarding understanding the virus better, what's the effect on the host cell, what's the genetics of the virus. And so there's lots of reasons here that we can cultivate animal viruses. They are very difficult to cultivate. And a lot of times what you do is you take a live animal and you inoculate the virus directly into the peritoneal fluid of the animal. This is done with small animals, mice, rats, hamsters, guinea pigs, and rabbits all the time. Okay. Sometimes for the production of like influenza virus, you can inoculate insects or insect culture. And sometimes if it's a research study, although this happens very, very rarely, the virus will be injected into a non-human primate just to understand what is the effect on the primate, because a primate is a good model of human physiology. For vaccine production, quite often bird embryos are used. Chicken eggs provide a very closed, essentially germ-free case. And as the embryo then divides and propagates, the virus divides and propagates along with it. Okay. And here's just this is an embryonic chicken egg. Shows the different cavities. I'm not so concerned that you understand the different places of the anatomy of a chicken egg, but you can see that depending on where the inoculation occurs, you have different places that are great for growing different viruses. Specifically the amniotic inoculation, that's the amniotic fluid around the developing chicken embryo, is good for influenza virus or mumps virus. You can go directly into the yolk sac for herpes simplex virus, the allantoic inoculation, which is the allantoic fluid that's outside of the amniotic sac, is good for the influenza virus, the mumps virus, Newcastle's disease, which is a disease where they make vaccines for chickens, and avian adenovirus, which is similar to bird flu. You can also propagate viruses in animal cells grown in culture. And what you do is you take a first round of culture after the cells are taken directly from an animal. So you might get an animal cell line. You could even do this with human cells, with like human foreskin cells. And then you propagate the cells in culture. They grow independently. And after the cells have been grown over several passages, then that will alter the chromosome number and morphology. So it wouldn't, you can't take these cell lines necessarily and go back and make a whole human from them or a whole animal, because the chromosomes have been altered. but then you can do this over long periods of time. And like flu vaccines are now routinely made from animal cell culture as well as insect cell culture. And the infected cells lice, and they form these regions of clearing called viral plaques. Okay, here is this is actually E. coli that's growing, and it was infected with a bacterial virus, a bacteriophage. And these zones of clearing is where the phage have eaten all the E. coli. Okay, and so they just form one dot at the middle of one of these zones of clearing, and they just eat their way out, and they eat their way out in a circular fashion, and each one of these circles is called plaques. And so if I want to find the area that's teeming with viral particles, then I'll just take a pipette or something that I can use to suck up some of the fluid, and I'll just suck up the fluid in the plaque, and it will be teeming with viruses. Okay. Medically important viruses. We have outbreaks of dengue fever, Rift Valley fever, yellow fever, rabies and Ebola are nearly always fatal, although there are some antibody therapies, what's called secondary or passive immunization, that are showing really, really good results for people who are already infected with Ebola. Polio and congenital rubella. Okay. And here is a vector that's loaded with virus. And this is yellow fever. Wonder why they call it yellow. Okay. Here's congenital rubella syndrome. This baby has cataracts. You can see the cataracts in the eyes. That is one of the characteristics of when a young infant gets rubella very, very young, or if the mother contracts rubella while she's pregnant. Okay. And I think I will leave it at that. We will cover prions in my next live in-person lecture next Tuesday. That would be October 10th. And that concludes this video lecture.